Hello, my name is Antti Vuolanto. I'm the CEO of Herantis Pharma. Herantis, we are a small listed company from Finland. We have ambition to stop the progression of Parkinson's disease, which is currently not possible with the current treatments. We have a lead asset called HER96, which is a small engineered peptide that truly has a unique mechanism of action with an easy route of administration. Our near-term milestones include next year running a clinical study to demonstrate the safety and blood-brain barrier penetration in men. The company has been around almost 15 years. We listed 2014, and since then we have been working with the same biology, but now we have this very unique HER96. But let's start describing the molecule by running a video. And in this video we see mice for motor coordination. And the first mouse here is an old, 18 months old mouse and easily walks through the bar. And the second one has an induced pathology, which is mimicking Parkinson's. And as you can see, it really struggles walking through the bar. And the third mouse is exactly like the second one, but has been treated with HER96 subcutaneously for 5 weeks. And as you can see now when they are running in parallel, the treated mouse is superior compared to the pathological mouse and even quicker than the normally aged mouse. And this is exactly what we want to transform from the preclinical studies that we have conducted into men and finally to the treatment of Parkinson's patients. So why Parkinson's disease is such a big problem currently? So Parkinson's disease is caused by the degeneration of dopaminergic nerve cells in the midbrain. And this degeneration results in decreased amount of dopamine that is needed for motor coordination. And the lack of dopamine results in the typical symptoms in Parkinson's disease, so motor symptoms like tremors or slowness of movement, but also non-motor symptoms such as dementia, depression, or constipation. The diagnosis of Parkinson's disease is based on the symptoms. So there is no prognostic diagnosis. At the time when the first symptoms appear, the patient goes into to doctor and then the diagnosis is made. At that time already, over half of the dopaminergic activity has been lost. So if you want to act on the disease, we need to be able to do that right after the diagnosis. The current treatments that have been around for almost 60 years, based on levodopa, they help to restore the dopamine level in the brain. However, they don't affect the nerve cells, so they don't stop the progression or even slow down the progression of the disease. And why is that? One of the main reasons is blood-brain barrier. That is a biological barrier or membrane that prevents pharmaceuticals and also other harmful or not other harmful, but harmful substances to enter the brain tissue. And actually, over 95% of pharmaceuticals, even the small molecules, cannot efficiently pass the blood-brain barrier. So there has been many promising therapeutic candidates that have worked in the preclinical settings, but unfortunately they haven't been able to be delivered to the brain, to the target tissue. And if you look at the market, currently there are approximately 10 million patients, Parkinson's disease patients, globally. And that is steadily and actually quite rapidly increasing. So in 15 years, it's been estimated to grow to over 12 million patients. Currently, the market size for the Parkinson's drugs is around 5 billion. However, it's been estimated to grow over $11 billion within the next 7 or 8 years, and that will be driven by the disease-modifying drugs that will hopefully enter the market. And now here comes Herantis HER96. We strongly believe that it has all the qualifications to really address this unmet clinical need to be able to modify the disease, to stop the progression of the disease. So why we believe that HER96 is a perfect drug candidate for Parkinson's disease? First of all, the design of the compound. It comes from a natural protein called CDNF. And the company has a long history in studying CDNF biology and its effects in Parkinson's disease. And we have even run a clinical study with CDNF with fairly good results. However, we were able to capture the activity of the CDNF protein into a small molecule, HER96, that mimics the active site of the CDNF protein. We have modified, so the HER96 is a modified peptide that resists the degradation in human body. It's manufactured very cost efficiently with chemical synthesis, and we have strong IP position there. But why it's so unique? So it has this mechanism of action that comes from the natural protein that has been shown to really powerfully protect the neurons. But it also very efficiently goes through the blood-brain barrier. So we can simply administer that with subcutaneous, so a skin injection. And what we expect that the Parkinson's disease patients then would benefit from HER96 once it's available for the treatment. So we believe that it can stop the progression of the Parkinson's disease at an early stage. So right after the diagnosis, HER96 treatment could be started, and that would stabilize the disease and keep the patients with good quality of life related to Parkinson's disease for the rest of their lives. We have easy patient-friendly administration, subcutaneous, such similar like we have for insulin injections in diabetes. And we have very good safety profile based on preclinical data. So how HER96 functions? So we have demonstrated that HER96 acts on the key mechanisms that results in the suffering of the midbrain neurons. So first of all, the left lower corner, we affect on the chronic neuronal stress. So we relieve the stress that the neurons experience. We directly affect on alpha-synuclein aggregates. They are toxic protein aggregates that accumulate into Parkinson's disease brain. And we also act directly into surrounding tissue neuroinflammation. And we have studied how HER96 does this, and we know that HER96 modulates the process called unfolded protein response pathway. So it controls the level of cellular stress in the neurons. And it in turn also results in control or modulation of proteostasis, so how the building blocks of our cells, so proteins, how they are synthesized, how they fold, how they traffic, and finally how they degrade in the cells. and thereby preventing the accumulation of unwanted misfolded proteins and protein aggregates. And through the action with unfolded protein response, it also stabilizes the environment in the surrounding tissue, so it affects on the neuroinflammation. We have studied HER96 in the best possible animal models. So we have an animal, an aged mice model where we have injected alpha-synuclein, these toxic protein aggregates, directly into the target area in the brain. And we have, in addition, added injection of a compound that makes the pathology actually progressive, exactly like Parkinson's disease. Let's not go into the details, but just to state that the mice represent Parkinson's-like pathology, and it's progressive. And here is a snapshot of the data that we have. So in the experiments, we initiate the pathology, we let that pathology to develop 1 week, and then we start dosing HER96 subcutaneously under the skin for 5 weeks, and then we analyze the animals. And now here in the right-hand side, we can see the data. So the disease animals are shown as a gray bar, and the treated animals are represented as the blue bar. And the top bars, they represent the dopamine neuronal survival in the midbrain. And as you can see, using HER96, increases the survival by 45%. The mid graph represents the amount of toxic alpha-synuclein aggregates. And as you can see, HER96 treatment decreases the toxic substances by 32%. And the lowest one represent the amount of neuroinflammation. And as you can see, HER96 treatment has basically removed the neuroinflammation completely. In addition to the mechanism of action, the second important thing is to be able to deliver the drug into the brain. And these we have shown in 3 different animal models: mice, rats, and dogs. So single administration under the skin, and then measuring how much of that dose has been delivered into the brain tissue. We know in rat experiments that actually 20% of the effective dose or the concentration is delivered to the brain, which is a very high amount. We actually exceed the therapeutic concentration 100-fold in these rats. And of course, this gives us the beliefs that when we go to the men next year, we truly can deliver enough drug to also Parkinson's disease brain in men just using a simple subcutaneous administration. So, what are the near-term milestones? So basically, even this year still, we are going to submit the clinical trial application for a clinical study that we will run next year. And the next year's clinical study will be a safety study. So we will recruit healthy volunteers with a single ascending dose study design. So of course, the primary endpoint will be safety. However, we will also have a separate cohort of elderly volunteers from whom we will collect CSF samples to measure the blood-brain barrier penetration of HER96. That, of course, is a major piece of information for us going forward then into clinical development. And we are fully funded through these milestones as we conducted a financing round earlier this year. What we aim is definitely to have a partner for HER96 in Parkinson's disease. So the development of drugs in neurological disorders, especially neurodegeneration and Parkinson's, that's time-consuming, requires a lot of resources, and as a small biotech company, we definitely would not be able to deliver such a drug into the market. So we are currently actively negotiating with potential partners, and of course, very difficult to predict when such a partnering deal could happen, but at least that's something that we are strongly committed to work towards as we speak. Okay, thank you. That's all that I had in mind here.
Okay, thank you very much, Antti, for the presentation. Let's start with a practical question. Your drug candidate passes the blood-brain barrier, or 20% of the drug goes into the central nervous system, where it then supposedly takes place. So can you give us an idea, is that a lot or not compared to other drugs that affect the central nervous system? Is that something that we should be happy with?
We are extremely happy about that. I would say that that's a very good number. Of course, it's difficult to compare different molecules because of their characteristics. If we go into antibodies, which are large biological molecules, their typical number is around 1%. But even I would say that the most relevant question is whether you will have a pharmaceutically active concentration. And we, as I said, we clearly exceed that by almost 100-fold in a rat model. So that's a very good sign for us.
Okay, and as you mentioned, you are administering the drug under the skin, but you also have some project to make the drug more soluble and maybe deliver it via oral route or by eating the drug. So what is going on there, and what is the role of that project related to this, your whole drug program?
Yeah. So basically, if you think about how we foresee HER96 in the treatment of Parkinson's disease. Once a patient, maybe my age or a 65-year-old person, would be diagnosed of Parkinson's, and HER96 treatment would start at that day, and then it would continue until tens of years. Having a subcutaneous injection like insulin, that's doable and that's fine. But of course, the preferred route would be just to have a tablet or capsule that you can take once a day or 3 times a week or whatever. So basically, for the present study that we are doing next year, and maybe until phase 2, late phase 2, subcutaneous administration would be just fine. However, if we think about commercialization, maybe a tablet would be even more preferred. So the development of a new pharmaceutical formulation is, I would say, it's important, but it's more like add-on to what we have. So it's not essential for us right now.
Okay, thanks. And then about the clinical program, so you plan to start it H1 or perform the first administration and then have some result on H2 next year. But is that will you also finish the phase 1 next year, or will it continue then forward? What is the timeline for the phase 1 study?
Yeah, the plan is to complete the study next year and to be able to report the top-line safety and blood-brain barrier penetration data by the end of the next year.
Okay, and as you mentioned, your part of your strategy is to look for partners. So how about your drug candidate's mechanism of action? It's a little bit untypical. So how has the response been, or in the discussion? Are there any thoughts in the industry about the mechanism of action?
That's a very good question. It's both good and bad that we have a truly very different mechanism of action compared to any other drug candidates currently. And it divides also the pharmaceutical companies. We have been discussing with 60 companies during this year, and some of them say that, okay, we are more interested about well-established mechanisms, but some of them are really enthusiastic about, well, this sounds really good because we need to have something new. There have been some consensus about, for example, antibodies have been developed for, I would say, at least a decade to decrease the amount of alpha-synuclein aggregates, but there hasn't been very good results so far. So actually, I would say that there is more enthusiasm currently with new mechanisms of action.
Yes, and maybe we can mention to the viewers that there are many drugs on the market where the mechanism of action is not really known. So
Sure.
It's not unusual. Now you're moving from the preclinical animal studies to the clinical studies. So what are your expectations? Now you have data on how the drug goes through the blood-brain barrier, and it would be easy to think that sure, now you just repeat it in humans, but mice are not men. So do you know from previous studies in the field, like how probable it is that if you see crossing of blood-brain barrier in mice, will you see that same effect in humans as well?
Again, very good question. It is a species-specific phenomenon, how efficiently the molecules goes through the blood-brain barrier. So we see different amount going in mice, different amount going in rats, and different amount in dogs. So we will definitely have very likely a different amount going in men as well. However, there are existing scaling factors, so in a way that's forecasting, and we have of course done that. So we have certain expectations how much we think we should see. However, the study design allows a rather big window around what we expect and to be able to detect. And as we know that even fairly low concentrations are enough for the pharmaceutical effect, we are pretty confident that we are able to establish the blood-brain barrier penetration also in men with the study design that we have.
Okay, final question. In your clinical trial, are you looking for any parameters of efficacy, or is it only about the safety and penetration of the blood-brain barrier?
Yeah, we are running the study with healthy individuals, so basically we can't assess any signs on Parkinson's disease. However, we have some biomarkers in the study which could demonstrate how HER96 affects overall in the biological systems, and we have identified some biomarkers that would then be hopefully be able to help us further on in Parkinson's disease patients that would then would be a follow-on clinical study once we have completed the next year study.
Yes, and as you mentioned, this is potentially a drug that would be used for a very long time, so that's also interesting as an investment case. So we will be following very with great interest how you will move forward. Thanks very much.
Thank you.